Hi everyone, this is my video on Discussion 2 Cytology. My name is Hannah. I just am going to go over the cell diagram and its organelles. So in the hierarchy of body organization, cells are the first to display characteristics of life. The basic components of a cell are the plasma or cell membrane, which we have right here, the cytoskeleton, and the cytoplasm. So let's start with the plasma membrane. So the plasma membrane surrounds the cell. It's made of proteins and lipids. Its function is to define the boundaries of the cell, govern its interactions with other cells, and control the passage of materials to and out of the cell. In addition to the cell membrane, many cells have surface extensions that aid in absorption, movement, and sensory processes. There are 4 different types of surface extensions. There are microvilli, there is cilia, which, oops, I missed that. Well, I'll just, cilia, flagella, and pseudopods. So microvilli serve primarily to increase the cell's surface area and are best developed in cells specialized for absorption, since increasing the cell's available absorptive surface will increase the cell's absorption ability. Cilia are hair-like processes present in nearly every human cell in the form of a single non-motile primary cilium, the function of which in some cases is a mystery, but apparently many of them are sensory. There are also motile cilia that aid in propelling mucus and other materials. There is only one functional flagellum in the human body, and it is the whip-like tail of the sperm that helps a sperm propel itself. Pseudopods are cytoplasm-filled extensions of the cell. They vary in shape and change continuously. An example would be white blood cells or neutrophils, which crawl around on finger-like pseudopods. When they encounter a bacterium or other foreign particle, they reach out with their pseudopods to surround and engulf it. So the next basic component of a cell is the cytoskeleton. The cytoskeleton is actually part of the cytoplasm, which I'll get to next. So the cytoskeleton is a network of protein filaments and cylinders that structurally support a cell. So the cytoskeleton determines a cell's shape, it organizes its contents, and it directs the movement of the materials within the cell, and also contributes to movements of the cell as a whole. The cytoskeleton is composed of microfilaments, intermediate filaments, and microtubules. So microfilaments, which are made of the protein actin, form a fibrous mat called the terminal web, which you can see me putting together here. It's also called the membrane skeleton, and it's on the cytoplasmic side of the plasma membrane. It is thought that without the terminal web, the plasma membrane would not hold together. Intermediate filaments are thicker and stiffer than microfilaments. They give the cell its shape, resist stress, and participate in junctions that attach cells to their neighbors. A microtubule is a non-permanent cylinder made of 13 parallel strands called protofilaments, which are long chains of tubulin. Microtubules radiate from the centrosome and hold organelles in place, help a cell maintain its shape and rigidity, and act somewhat like railroad tracks, which aids motor proteins in transport within the cell. So the next component of a cell, which is actually a majority of the content that is in this video, is the cytoplasm. So the cytoplasm is found between the nucleus and the surface membrane. So let me just grab the nucleus here first and get that. All right, so there's our nucleus. So the cytoplasm is found between the nucleus and the surface membrane. It contains the cytoskeleton, the organelles, and inclusions, as well as the cytosol or intercellular fluid that they are embedded in. So here's our nucleus. It's the control center of the cell. It is the largest organelle and contains the cell's genetic information. Most cells have one nucleus, but there are a few exceptions. Mature red blood cells are anuclear, meaning they have no nucleus, and a few cell types are multinucleate, meaning they have anywhere from 2 to 50 nuclei. Examples of multinucleate cells include skeletal muscle cells and some liver cells. The nucleus is surrounded by a membrane called the nuclear envelope, which I am putting right here. So the material within the nucleus is called the nucleoplasm. It contains chromatin, which is composed of DNA and protein, and one or more nucleoli where ribosomes are produced. So the next organelle is called the endoplasmic reticulum. It's a system of interconnected channels called cisternae, and it synthesizes steroids and other lipids, detoxifies alcohol and other drugs, and manufactures all membranes of the cell. There are two distinct areas of the endoplasmic reticulum. The rough ER gets its name from the ribosomes that are present. It's most abundant in cells that synthesize large amounts of proteins, such as antibody-producing cells and cells of the digestive glands. And then there's the smooth ER, which is abundant in cells that engage extensively in detoxification, such as liver and kidney cells. And the rough ER is continuous with the outer membrane of the nuclear envelope, while the smooth ER is continuous with the rough ER. So although they are different, they are part of the same network. Ribosomes are small granules of protein and RNA found in the nucleoli, in the cytosol, and on the outer surfaces of the rough ER and nuclear envelope. They read messenger RNA and assemble amino acids into proteins specified by that code. The unattached ribosomes scattered throughout the cytoplasm make enzymes and other proteins for use within the cell. When a ribosome makes protein destined to be packaged in a lysosome or to be secreted from the cell, it will attach itself to the rough ER. So the next organelle we have is the Golgi complex. It's a small system comprised of flat sacs called cisternae, which are responsible for synthesizing proteins, as well as putting the finishing touches on newly synthesized proteins received from the rough ER. Finished proteins are packaged in Golgi vesicles and then are transported within the cytoplasm. Some vesicles become lysosomes, which I will go over next. Some migrate to the plasma membrane and fuse with it to contribute fresh protein and phospholipid to the membrane, and some become secretory vesicles that store a cell product for later release. Examples of this would be breast milk or digestive enzymes. So here we have a lysosome. The primary function of a lysosome is to hydrolyze proteins, nucleic acids, complex carbohydrates, phospholipids, and other substrates. Lysosomes are also responsible for autophagy, which is when a cell's surplus or non-vital organelles and other cell components are digested and disposed of in order to recycle their nutrients to more important cell needs. So peroxisomes are produced by the endoplasmic reticulum. And the general function of a peroxisome is to use molecular oxygen to oxidize organic molecules. So this reaction produces hydrogen peroxide, which is then used to oxidize other molecules. And the excess is broken down to water and oxygen by the catalase enzyme. Peroxisomes are especially abundant in liver and kidney cells as they neutralize free radicals and detoxify alcohol, other drugs, and a variety of bloodborne toxins. They also decompose fatty acids into 2 carbon fragments that the mitochondria use as an energy source for ATP synthesis. Proteasomes are hollow cylindrical complexes of proteins located in both the cytoplasm and the nucleus. Their function is protein disposal, so a cell will tag undesirable proteins such as ones that are damaged and non-functional, or foreign proteins introduced during a viral infection. These tagged proteins are transported to a proteasome, which will unfold and break it down into short peptides and free amino acids, which can then be used to synthesize new proteins or presented to the immune system for further degradation. Proteasomes degrade more than 80% of a cell's proteins. Mitochondria, right here, also known as the powerhouses of the cell, are responsible for synthesizing ATP. A double membrane surrounds each mitochondrion. The inner membrane usually has folds called cristae. The space between the cristae is called the matrix and contains ribosomes, enzymes used in ATP synthesis, and mitochondrial DNA. It's important to note that energy is not made in the mitochondria. Rather, enzymes located on the cristae extract energy from organic compounds and transfer it to ATP. Centrioles are composed of a circle of 9 triplets of microtubules. 2 centrioles lie perpendicular to each other within the centrosome, which is a small clear area of the cytoplasm near the nucleus. Pairs of centrioles play a role in cell division, while unpaired centrioles form basal bodies of cilia and flagella. So the final component of a cell are inclusions, which are accumulated cell products from cellular metabolism and foreign matter retained in the cytoplasm but never enclosed in a membrane. And they are not essential to cell survival. So there you have it. There is my cell structure model and review. Thanks so much for watching, and I'm looking forward to seeing your videos. Thanks.